Well, good morning. Thank you so much for the invitation. I'm really excited to be joining for this seminar series. I am a new faculty member in the Department of Chemistry and Biochemistry and the SCAG School of Pharmacy and Pharmaceutical Sciences. And I lead a chemical biology group that's really focused on using proximity pharmacology, and I'll explain what that is, to tackle questions we were previously unable to answer with traditional small molecule inhibitors in both neuro-oncology and neurodegeneration. And so today I'm going to be talking about some of my postdoctoral work I did to understand targeted protein degradation pharmacology, build a map of tractable targets across the kinome, and also expand our work to uncover misfolded proteins commonly found in neurodegenerative diseases and proteinopathies as a new class of tractable targets. And so I will get started. I'm super excited to be here. And today I'm going to talk, briefly introduce the kinome as a target class and explain how we think about protein classes or target classes in drug discovery. I'll then introduce the concept of targeted protein degradation and our methods for building an experimental map of the degradable kinome. In part two, I'll expand on this, really talking about how we examined the cellular and chemical variables affecting targeted protein degradation outcomes and what we can learn from this as medicinal chemists and small molecule drug hunters to design better approaches to uncovering small molecules that are selective, that are potent, and they have the activity profile that we want, and how we can use multi-targeted degraders as chemical tools to interrogate the ubiquitin proteasome system. Finally, I'll show how our curiosity as to the limits of this technology led us to thinking about whether we could go after so-called undruggable targets and how we used our knowledge of targeted protein degradation to develop small molecules that can selectively clear the oligomeric forms of tau found in frontotemporal dementia. So kinases, as many of you may be familiar with, are a highly druggable gene family involved in virtually all signal transduction pathways in the cell, making them excellent targets in drug discovery. And there are over 40 approved kinase inhibitor drugs in the clinic, currently mainly in oncology and limited to a few kinases, but a large number of other kinase targets and a large number of other indications that are where kinase drugs look promising are currently in phases 1 through 3, including, of course, neurodegenerative diseases. And one of the reasons why kinase-focused drug discovery has been so successful is that kinases have a shared substrate, a small molecule called ATP. And one successful method for kinase inhibitor discovery has been to take this gene family-based approach where we synthesize small libraries based on privileged scaffolds that harbor a pharmacophore that can bind to the conserved structural motif. In the case of kinases, this is the hinge binding region, and create profiles of our kinase binders using biochemical or cellular assays. What this means is once we identify a desirable target, whether that's through a collaborator who's identified it through mechanistic studies, whether that's through genome-wide approaches, or reading of the literature, we can then go to our library, figure out the initial structure-activity relationships, and do medicinal chemistry on our identified leads to improve potency, selectivity, and pharmacokinetics. And the goal of all of this is to do either target validation or to do biological characterization to understand the role of that kinase and that kinase activity in the cellular context. And labs such as my postdoctoral lab, which is Nathaniel Gray's lab at Harvard Medical School, and my own lab here at UCSD that develop these kind of molecules, usually make them open access and provide them to the academic community for use in perhaps disease modeling labs that don't have access to their own synthetic chemistry. And as a postdoc, I was privileged to work on many of these exciting projects, including some of those shown, well, including all of those shown on the screen and a few others. So we developed first-in-class ligands for understudied kinases, including DCLK1 and 2, which is involved in neural development, ERK5, CDK14, and the lipid kinases PIP4K, alpha, beta, and gamma, as well as highly optimized kinase inhibitors with improved selectivity and scaffold diversity for identified kinase targets. So I was based at Dana-Farber Cancer Institute. That was a big disease focus of the lab, such as Aurora kinase A, B, and C, and PI3K delta gamma. And so what we found with some of these projects is that although these chemical tools, which are highly selective, were incredibly useful for looking at the downstream effects of kinase inhibition and selective kinase inhibition of these targets, sometimes our selective small molecule probes were not giving us the same phenotypes as the genetic knockout studies that had been used to identify them as interesting targets. And so we were really excited to hear about and see the development of targeted protein degradation as a new type of small molecule in the labs of Craig Cruz, Jay Bradner, Alessio Trulli, and others. And so this chemical strategy allows us to overcome this limitation. So targeted protein degraders, as you can see on this screen, are heterobifunctional small molecules that on one side contain a binder to the substrate adapter component of an E3 ubiquitin ligase. This is linked via a short chemical spacer to a binder of your target of interest. And so upon addition into a cell, this small molecule brings your target in close proximity with the ubiquitin proteasome machinery, triggering its polyubiquitination by the bound E2 enzyme over here and subsequent proteasomal degradation. And so if we compare this to other approaches that you may be more familiar with using, it has the dosage control of pharmacological inhibition, but it affects protein abundance. So this is a really cool way of engineering the proteome with all the precision of genetic approaches, but the fast kinetics and reversibility of small molecules. And it can be incredibly selective. And one of the things that targeted protein degraders can do that small molecule inhibitors cannot is really target the scaffolding functions of proteins such as kinases, which often have many other domains. And as we'll talk about later, it also has the potential to expand the druggable target scope. And so when we're developing these molecules, their pharmacology is actually very different to inhibitors. And so inhibitors are occupancy driven. The drug concentration controls the pharmacodynamics in vivo. Whereas for degraders, these are event driven molecules. So they trigger catalytic turnover of your target of interest. And it's actually the protein resynthesis rate rather than the actual small molecule occupancy that's controlling the pharmacodynamics in vivo. And so when as chemists, like myself, we think about developing these molecules, we not only have to think about optimizing target binding, but optimizing their ability to dimerize these two proteins of interest and trigger ubiquitination. These molecules are larger than many traditional drugs, and so we have to think carefully about cell permeability and the duration of target degradation. And because the presence of the E3 ligase and its activity are absolutely required for this mechanism of action, there's also the potential for cellular and organismal selectivity of degradation, and that's something you may or may not want and may have to tune into your compound. And of course, there is a disconnect between pharmacokinetics and pharmacodynamics that we just talked about. So with all of this in mind, we had built a suite of biochemical assays to understand degradation pharmacology and had started making small molecules against a variety of kinase targets. However, what we found in our early work was going from this beautiful scheme to a potent fast-acting degrader with a desired selectivity profile, no simple task. And given the fundamental differences that we just talked about with inhibitor design and the relative newness of the field, perhaps this was unsurprising. And so although negative results are rarely published, this was an observation that had also been made by other leaders in the field. And so you can see here, it's been put very elegantly as design elements for successful PROTACs. That's another word for targeted protein degraders. Base drugs are currently being elucidated and then being translated on the blog In the Pipeline, which is led by Derek Lowe, the head of chemical biology at Novartis, as what this means for medicinal chemists like myself, which is that this field is empirical and you really have to try a lot of molecules. And the reason for this is that each element of a degrader, from the target binder, the attachment chemistry, the linker, and the E3 ligase that you recruit can have a dramatic effect on the compound's activity. And there may also be cell-specific and target-specific variables that can play into that as well. And so this resource-intensive discovery phase of targeted protein degradation campaigns has been highlighted by leaders in the field as a major limitation for its broad approach. So our initial efforts to identify low-hanging fruit for TPD within the kinase family took advantage of highly multi-targeted kinase inhibitors, such as desmethoxy AG0624, which again have these hinge binding motifs. And this is shown on the left, to create multi-kinase degraders. And so you can see here that this compound is able to bind to over 200 kinases biochemically. This is a kinome scan plot. And a talented graduate student in the Gray Lab, Hubert, treated 2 cell lines with these compounds and was able to identify around 25 degradable kinases. So armed with this knowledge, the Gray Lab and others were able to rapidly generate potent and selective degraders for these targets. And so our hypothesis that these targets were highly amenable to this technique seemed to be validated. But what happens if your kinase of interest is not in this kinase list? You can see here a lot of cell cycle kinases that might be relevant to oncology, but maybe a little bit less relevant to, for example, neurodegenerative disease. How do we hack this problem? Well, from our experience creating inhibitors across the kinome, we decided to take a target class-based approach and build an experimental map of the degradable kinome. And so our goals were to answer some of the outstanding questions in the field, such as how do we prioritize degradable targets? Which binder is the best one to choose for degrading my target? Is this the same as the most potent binder? How do we rapidly find and optimize our leads? Which E3 ligase is the most compatible? And many others. So we want to answer these questions with statistical rigor across an entire target family, because what we had seen is that some of the observations were not holding true when we tried to apply them broadly. So what we did, we built our kinase degrader library and pre-screened these compounds for cell permeability in E3 ligase cellular target engagement assays. Based on cell permeability and scaffold diversity, we selected 91 of them for profiling using mass spectrometry-based global proteomics analysis and used short time points to monitor direct degradation effects and minimize detection of downstream or secondary effects. We were then able to identify hits, understand degradability, and also generate hypotheses that could then feed into our synthesis and allow for systematic variable exploration. So this is our library here. You can see it's composed of many different chemical pharmacophores, including type 1, type 1.5, type 2, and allosteric binding modes, a number of different PEG-based linkers. And in this case, we're only looking at recruiting the cereblon and VHL E3 ligases. And so this would be a really interesting exercise and experiment to repeat with some new E3 ligase binders. And I'll talk about that a little bit later on. So we calculated using published and in-house data that the parental inhibitors contained in this library bind to 93% of the DiscoverX kinome scan panel. So that corresponds to a minimum of 70% of the human kinome. So we have great coverage in terms of kinase binding. What we're excited to find is through these experiments, we identified 172 degradable protein kinases from all clades of the kinome tree. Reassuringly, we rediscovered the vast majority of degradable kinases reported in the literature. And we also looked beyond the 518 human protein kinases at the extended kinome, which is metabolic kinases, lipid kinases, and pseudokinases. And this has been compiled recently by the Sorga lab in this nice preprint listed on the bottom of the slide. And so we were able to observe degradation of 40 of these proteins by compounds in our library, bringing the total above 220 kinases that were able to degrade and provide lead molecules and whole protein profiles for. And so what we also wanted to use our database for is to understand the relative tractability of our targets. This not only validates and increases confidence in our hits, but also tells us something maybe about the intrinsic ability of these kinases to be targeted by this specific type of small molecule. And what we found was that there were a number of kinases that were degraded by a large number of different degraders, so downregulated in multiple experiments. And correcting for unique compounds, we were able to assign every kinase a degradability score based on the number of unique molecules that could degrade it with statistical significance. We wanted to know if this truly was telling us something about a kinase's propensity to be targeted, or if we were simply reproducing the highly studied kinome, which has been extensively targeted by kinase-directed small molecule medicinal chemistry campaigns. So to do this, we looked for correlation with different knowledge metrics, such as the number of publications in PubMed, the number of citations, the number of structures in the PDB, and so forth. And we're pleased to find no correlation. And so given this information, we actually felt that our library was relatively unbiased, and we were able to search for hits against the NIH's understudied kinase list. And so many of these understudied kinases are actually relevant to neurology and neurodegeneration. But this list is defined as kinases whose function is poorly understood and for which there are no selective chemical probes available. And so we identified degrader hits for 16 understudied kinases, including many attractive starting points for beginning a selective small molecule degrader development project, such as the example shown here, which degrades CDK17 and only 2 other kinases. And so in the next couple of slides, I'd like to share some examples to illustrate how this data set that we created can facilitate entry into targeted protein degradation projects. So in the first example, we use our frequency with which a kinase is degraded by unique compounds across the database as an indicator of its degradability, and identified Aurora kinase A as a highly tractable target. So synthesis of degraders based on a selective Aurora A inhibitor led to rapid identification of potent and highly selective Aurora A degraders, such as DOK4, which is shown on the slides. And you can see here through the whole proteome profile that Aurora A is really the only significantly downregulated target, and then a little bit of degradation of Aurora B observed as well, but that's right on the line of significance. We're able to validate that these degraders were potent and were able to effect complete degradation at 125 nanomolar concentrations as fast as 4 hours, and this was sustained out to 24 hours. We also see distinct pharmacology. And so we tested them in the MM1S cell lines and multiple myeloma line, which is resistant to alisertib, the parental inhibitor, but we can see a clear loss of viability caused by our Aurora degrader. So that's one case where you might be looking for a target which is low-hanging fruit, but much more likely you already have a target in mind from your biological studies, and you would like to develop a degrader molecule with which to study it better, validate it as a target, or perhaps as a therapeutic lead. And so in this second example, we decided to pick a kinase for which there are no selective small molecule inhibitors reported, and we picked the kinase CSK, which is involved in STING pathway signaling. And so it would be very unclear where to begin if we wanted to design a selective degrader based purely on the literature and not on our experimental data set. However, we were able to identify numerous compounds that could downregulate CSK at the 5-hour time point. And examination of the proteome-wide profiles of these molecules revealed really attractive starting points for medicinal chemistry development, such as this compound DB3291, which is based on dasatinib. And so CSK is not the most potent target of dasatinib, and dasatinib is actually a highly multi-targeted kinase inhibitor. So we'd never have thought to start here if we were simply reading the literature, but it's a nice example of how this data set can really accelerate targeted protein degradation research and in areas outside of oncology. So 220 projects is way, way more than any lab or labs can take on. And so we wanted to facilitate use of this resource to identify leads for kinase of interest, broadly accelerate kinase degrader discovery programs. And so we created a web tool that actually allows you to search by target of the compound or by small molecule structure and create your own heat maps, plots, change the cutoffs, etc. And there are a large number of hits, over 220, including many relevant to neurodegeneration, RIPK1, SARC, etc. And so I really hope people will use this resource. All the small molecules are also available on request from the Gray Lab. And so you can actually find a compound for your target of interest, get those compounds and test them in your own experimental systems. However, once you have an interesting lead compound, the next step is to optimize it. And as targeted protein degradation is a relatively new field, although there are many emerging guidelines and observations that have been reported, sometimes they're contradictory, and it's really hard to understand how broadly applicable they are, given that these studies are usually limited to one chemical scaffold, one protein target, and one cellular context. And so we wanted to get general conclusions about the frequency, magnitude, and significance of some of the new hypotheses in the field. So to do so, we identified 4 degraders from our collection that could together induce degradation of the largest number of unique kinases, allowing us to survey over 100 kinases in just 4 cellular treatments. And we use these to interrogate different effects of chemical and cellular variables. So the first thing we looked at was cellular target engagement versus degradation. We already know that those two are highly uncoupled due to the catalytic mechanism of action, but we wanted to understand just how much cellular target engagement do you need in order to have an effective degrader. And so in order to do this, we performed global proteomics analysis and also performed cellular target engagement across the kinome using the chemoproteomic profiling technique kinated. And I'm showing the results for just one compound on the left here, where we're plotting percent inhibition of binding versus relative abundance fold change. And we can see, and summarizing for all 4 compounds on the right, we can see that there is often no detectable target engagement, but we can clearly see degradation. And the converse, we can sometimes see target engagement and no degradation, showing us that target engagement is really not the crucial determining factor for targeted protein degradation. So there's been a lot of discussion about enabling the formation of that ternary complex between the target of interest, the small molecule, and the E3 ligase substrate adapter. So we wanted to evaluate that relationship. To do so, we performed matched experiments evaluating degraded targets using TMT-based global proteomics analysis in wild-type HEC293 T cells and evaluating complex targets using affinity purification mass spectrometry in cells expressing flag-tagged cereblon. Again, what we found was evidence of both productive ternary complex formation. And so this is one example compound shown on the left, and this is a summary on the right of what we observed across all 4 of those molecules. So we observed productive and non-productive ternary complex formation, where sometimes proteins were able to be complex, but they were not degraded. We didn't observe enrichment by our affinity purification mass spectrometry of many of the degraded kinases, indicating that formation of a stable, highly abundant complex is not a requirement, but rather transient or low abundance complexes that are highly catalytic can lead to effective degradation. So really importantly, another variable that can affect both complex formation and degradation is the cellular context. So we evaluated the target profile of our 4 selective degraders in 3 different cell lines of different tissues of origin, HEC293T, MALT4, and Kelly cells. And we were surprised to observe significant differences in the target space for the same compounds in each cell line. So encouragingly, many hits were overlapping, but some others were not, despite being quantified in all 3 lines, really highlighting the importance of characterizing degraders in different cellular contexts and the relevant cellular contexts of interest. And we'll see that this is important later on when we discuss targeted degradation of tau. So finally, the compatibility of the induced protein-protein interaction surface between the E3 ligase can also affect the degradation outcome. And so to understand how changing the recruited E3 may alter the target profile of our multi-targeted degraders, we synthesized matched pairs while we varied only the ligase recruiting component. And so I'm showing one example of matched pairs on the left, where we can clearly see that there are some kinases which are amenable to degradation by both small molecules, and this is fantastic. It gives us a lot of flexibility. There are some that clearly prefer one ligase over the other. And so when we look across all of the matched pairs that we synthesized and tested, we can see that by increasing the number of ligases that we recruit by only one, we're able to increase the target space by 20%, really indicating the target scope can be expanded even further by efforts to discover ligands that recruit new E3 ligases. Finally, we looked at linkerology, investigating how the linker, length, and exit vector affect the target profile, and we're able to identify kinases which were relatively resistant or robust to variations in the linker as those that were also identified as highly degradable across our entire data set, indicating that the plasticity of the ternary complex form may be an important feature of highly degradable targets. Finally, another aspect of target specificity that's amenable to manipulation of the linker is the degradation of common IMID targets, which is a consequence of using IMID molecules such as thalidomide to recruit cereblon. And so direct comparison of the levels of known IMID targets in response to these degraders provided some insights into the linker structure activity relationships. And importantly, we were able to look across the database to identify preferred attachment chemistry for rational design, polypharmacology, degrading zinc finger transcription factors, as well as kinases, or highly selective degraders that do not hit those IMID off targets. So we now have all of these multi-targeted tools, and we've been able to use them to understand our medicinal chemistry approaches and design better degraders, but what about using them to interrogate the ubiquitin proteasome system itself? We were curious about the role of unfoldases, in particular the ATPase P97 for degradation. So we treated cells with either degrader alone or degrader plus P97 inhibitor and observed rescue of degradation of most kinases. We validated this was not due to P97 inhibition alone by performing a time course experiment with just P97 inhibitor. And taken together, these results suggest the role of P97 in the handover of substrates to the proteasome goes well beyond its reported roles in terms of extraction of large cellular structures, but also includes unfolding of soluble polyubiquitinated proteins, such as the diverse array of over 100 kinases that we've looked at here. And so in our initial part 1, we've created this open access resource of degrader hits and whole protein profiles for over 220 kinases, quantified the effects of cellular chemical variables of TPD on outcomes across the kinome, and used multi-targeted degraders as tools with which to interrogate the ubiquitin proteasome system. But there was one really big question about targeted protein degradation that had been really highlighted but never proven, which was, can targeted protein degradation expand the druggable proteome? And so this led us to thinking about misfolded proteins that occur in neurodegenerative disease and whether we could target them using a protein degradation approach. And so for the next part of the talk, I'll talk about our efforts to identify targeted protein degraders of misfolded tau. So probably don't need to describe to this audience here, but in healthy cells, tau is intrinsically disordered soluble protein that binds to and stabilizes microtubules. And in disease, it undergoes abnormal levels of post-translational modification, including hyperphosphorylation, which causes it to dissociate from microtubules and aggregate into filaments and neurofibrillary tangles. And so the levels of these aggregates found in postmortem brain correlate with disease progression, but the relationship between pathology and phenotype is not clearly understood. And so although there are no small molecule drugs directly targeting tau, we do know how to create compounds that selectively bind to the disease-associated tau in form of PET tracers, which are used to detect the presence of these aggregates in the brain. And so one of the commonly used PET tracers, Talvid or T807, is used to stage patients which do not have amyloid pathology, but do have tau pathology, such as FTD. And so this only binds to tau. And so what we can see here are postmortem brain slices where we can see clear binding in A, which has tau pathology, no binding in B, which has amyloid pathology, but no tau pathology, and no binding in C, which is a brain from a patient which was not diagnosed with dementia. And so this molecule has selectivity over other aggregated proteins and over the soluble healthy forms of tau. So we sought to use TPD approaches to transform this binder into a molecule which actually had functional pharmacology. And you can see here it's used to correctly stage FTD patients. So what we did was we created or synthesized a degrader library where we took our tau ligand, varied the chemical ligand, and conjugated it to an E3 ligase ligand. And so what we were hoping was that we would be able to spare soluble tau and degrade only pathologically associated forms of tau. We focused on pomalidomide analogs predominantly, although we did try other ligases based on their expression profile in neurons and in the brain. So of course, to test these molecules, we cannot do what our colleagues, our admired colleagues who developed the PET tracers did and use postmortem brain, because we need an active ubiquitin proteasome system. So we turned to collaborators at Mass General Hospital, Steve Haggerty and others, who had developed these models of FTD patients. So clinicians at MGH identified a patient who had T807 positive PET scan, and they harbored a rare mutation in the tau gene, which is associated with a high risk of early onset FTD. And so we took skin punch biopsies, created dermal fibroblasts, which were reprogrammed into induced pluripotent stem cells, isolated neural progenitor cells, and then these neural progenitor cells could be differentiated into ex vivo neurons with physiological tau expression levels that recapitulate the genomic background associated with tauopathies. And this is all work done in Steve's lab at MGH. And so if we just compare the tau species in this FTD model to those in neurons derived from an age-matched healthy patient, we can see that this model shows accumulation, redistribution, and decreased solubility of phospho-tau species relative to healthy patient neurons at up to 5 weeks after differentiation. So we thought this would probably be a suitable model for testing our compounds, and we hope that these species would still bind to those tau PET tracers. And not shown here, these neurons also have defects in proteasome and autophagy pathways, which recapitulates disease and perhaps is a higher bar than cell lines for targeted protein degraders. So we tested our degraders in 8-week differentiated neurons that model early stages of FTD pathology and identified lead compound QC01175. And so what we can see here is that when we treat with our compound for 4 hours, we see reduction in total tau and importantly, high molecular weight phospho-tau, which is detergent resistant. We synthesized a negative control compound where we've removed a carbonyl from this glutaramide ring, which renders it unable to bind to and recruit to cereblon. And we showed that this compound has no effects on, no effects on levels of either tau species consistent with the proposed mechanism of action. So to evaluate if our degrader was broadly active against other disease-associated forms of tau, we tested our lead compound in neurons harboring a P301L mutation. And we're pleased to find it was still active at degrading total and high molecular weight phospho-tau in this context. So we wanted to perform a variety of mechanistic controls. We examined if we could isolate the ternary complex that was forming between tau and the CRL4 cereblon complex by pre-treating cells with proteasome inhibitor carfilzomib to block tau clearance, and then treating with vehicle negative control or our active compound, and immunoprecipitating for DDB1, a component of this complex. So we're pleased to pull down tau only in the presence of our active compound, validating our mechanism of action. We also performed rescue experiments, including proteasome inhibition, CRL activation, and binary competition, all of which confirmed our compound was acting through the mechanism we thought it was. And I'm going to skip over this in the interest of time to our proteomics. So we were interested to see, perhaps some of the chemists in the audience or even those familiar with pharmacology will see that we've used this really minimal tau binding pharmacophore, which is just this tricyclic molecule that may potentially bind to many other proteins. So we were curious to see if our compound was degrading other targets. So we treated neurons with 1 micromolar degrader for 4 hours and actually observed very limited off-target degradation, showing only these zinc finger proteins, which are neo-substrates of the cereblon pomalidomide complex. And using our knowledge from our degradable kinome project, we were able to engineer out that off-target activity in later series of molecules. We saw no targets in our negative control samples, and you may notice that we don't see downregulation of tau here, as the harsh conditions required to solubilize the tau found in these neurons were incompatible with our proteomics workflow. So this is purely an off-target screen. Having established proteome-wide selectivity profile, we really wanted to know, do our degraders maintain their selectivity for disease-associated forms of tau over healthy tau? And this is really important because this really gives us a complementary technology to gene editing, where of course you can only get rid of all of tau and you don't have that conformational selective control. So we expanded our testing to a total of 3 control lines of age-matched healthy neurons. One example is shown on the right here, and consistently saw no reduction in tau, confirming that the conformational specificity of the parental T807 ligand had been retained. And so with this well-characterized selective degrader in hand, we used our compound to assess the phenotypic consequences of tau clearance in these FTD-associated neurons. So these FTD neurons are uniquely vulnerable to cellular stresses such as ROS stress, mitochondrial stress, and including the proteotoxic amyloid beta peptide, which is present in the brains of Alzheimer's patients. And so we can see that this stress vulnerability is not present in healthy neurons. And so you can see on the left here, this is a viability curve at 24 hours, where in orange and red are FTD neurons, which are derived from patients and have those tau mutations, showing clear loss of viability after treatment with this peptide. However, this is not observed in aged match healthy controls, shown in black, and can also be rescued by knockout of the tau gene at the iPSC stage. So we were curious as to whether clearance of this misfolded tau would be able to rescue the cell stress vulnerability of our neurons, or whether it had already caused irreversible damage. So what we did was we took 8-week differentiated neurons, we pre-treated them with either our tau degrader or vehicle control, and then we treated them 8 hours later with a cellular stressor such as high-dose amyloid beta peptide, and at 24 hours observed the effect on viability. So in the absence of our cellular stressors, we can see that our compounds alone have no effect on viability, they are non-toxic. And we set our level of vehicle and amyloid peptide such that 50% loss of viability was observed for the neurons at 24 hours, so we could see both increases and decreases in viability. We were really pleased to see rescue of viability loss or cell stress vulnerability upon treatment with our degrader compound and not with our negative control. And we're happy to see that the level of rescue was almost comparable to tau knockout cells that have never seen this folded tau. And so we thought this was an exciting result for our initial compound. And so just to wrap up, targeted protein degradation is a really broadly applicable new strategy for clearing liganded targets from the cell. And large-scale chemoproteomic and pharmacological approaches can be used to reveal fundamental principles of degrader pharmacology and accelerate degrader discovery. We then identified misfolded proteins as a new class of druggable targets amenable to targeted protein degradation. And my lab at UCSD is really interested in taking a target class or target family-based approach, given the number of PET traces and generic aggregate binders that are out there, to really understand this class as a new family of druggable targets, to create chemical tools to understand targeted degradation and targeted clearance via multiple different pathways, not just the proteasome. And to really get at that question of what are these misfolded proteins doing at various stages of diseases and different proteinopathies. And so super excited to be connected with the Alzheimer's Center, yeah, really excited to be starting my lab at UCSD as well. Obviously, this isn't work I did all by myself. I'm predominantly a medicinal chemist, and so there was a ton of help from especially proteomic scientists in the Fisher lab, of course, our FTD scientists in Steve Haggerty's lab at Mass General Hospital, as well as a whole bunch of other collaborators and funders. And this is also a really big team effort from the Gray and Fisher labs in particular, which there are so many of them, I resorted to having pictures. And so again, thank you so much for your attention, and thank you so much for the invitation to talk in the seminar series. I'm really happy to take any questions.